Good evening, folks. I'm very happy to be back with you again, sort of, kind of. The Zoom stuff is, I think, less than optimal, but it's better than nothing. But I hope maybe in the future to be with you in person. So let's get on with this. This is a talk entitled Valuable and Vicious Viruses and a Vaccine. So first, I'm giving a disclaimer that what I have to say in this presentation is not necessarily the policy or viewpoint of Arizona Christian University, but it very well may be. So first, let's talk about the position of viruses in the biological spectrum. And when we talk about viruses, we also have to do a lot of talking about bacteria as well, because the interaction between bacteria and viruses is critical in the big picture. So viruses can infect every type of cell, but I have to say not any virus can infect any type of cell. A particular virus will be able to infect a particular type of cell. And we're not to think of viruses as living things, since they are obligate intracellular parasites. Now, in plain English, what that means is that viruses have to enter into living cells, bacterial cells or cells of animals or people or plants, in order to be able to reproduce. They are basically collections of proteins that only can, and DNA or RNA that can only reproduce if they invade a specific host cell. And so they're more than lifeless molecules, but they're not living. So they're in this gray zone in between. So it's probably best to think of them as infectious particles, because they are highly contagious. Now, to give you an idea of viral size, first I'll just mention two bacteria here. On the left, you see the round one. That would be a representative of Streptococcus, which is the common cause of bacterial sore throat. We call it strep throat for short. And then you see the E. coli on the right-hand side, which belongs in our gut. It's a normal inhabitant of the intestine, the small intestine, and the large intestine also. And you can see it's about twice as long as the Streptococcus. And you see there that their measurements are 1 micrometer and 2 micrometers. So a micrometer is a millionth of a meter. And you see here in that arc along the bottom of the curve there, different types of cells or yeast or viruses arranged by size. And you see there, number one, which is a large virus. It's 2/10 of a micrometer. And then at the other end of the scale, you see a yellow fever virus, which is 22 thousandths of a micrometer. So it's extremely small virus. And then here you see at number 3 is a rabies virus, but it's also the same size as the COVID-19 virus. So to help relate that to real life things, a piece of paper here, ordinary copy paper, or an average sized human hair on the bottom right, are about the same thickness, about 10 micrometers. So you can see here that the COVID virus is about 1/8 of the thickness of a piece of copy paper. So that's maybe a good way for you to be able to relate to something in everyday life, as to the size of the thing. So it's a pretty small particle here. Now, these viruses are in 4 basic shapes. So at the top left, you see a bunch of spheres. And then top right, you see a rod, a cylindrical rod. Then bottom left, this is hard to make out, but it's a 20-sided figure. It has 20 actual sides with corners. And then the bottom right, you see one that kind of looks like a lunar module lander there. And those are called bacteriophage. That class of viruses attack bacteria, and the word bacteriophage means bacterial eater. So here's a sharper picture of the bacteriophage with an electron microscope. And you can see there it is in a diagrammatic fashion, and it does bear resemblance to a lunar lander. So why don't we take off and get into more gory detail. So first we'll talk about the evolutionary ideas of how viruses came into being. But first I have to relate this to a bacteria. So this is a generic diagram of a bacterium. So it has its either DNA, in this case the bacteria, it would be DNA inside. There's cytoplasm, it has the various molecules and important metabolic pathways inside the shell, the multi-layered shell, which is made up of several different layers of different types of material, and then some flagella or whips, which would be used to move it through the fluid that it's located in. So there are three basic theories that the different evolutionists have as to how viruses came into being. The more popular theory is that they think that first living thing was a bacterium, which they cannot explain how it came into being, but that with time, certain bacteria lost various parts and became a virus. So you see here the generic bacterium, and then you see what viruses actually have. So inside is either DNA or RNA, and then a protein coat, which is called a capsid. So the capsid is a protein coat surrounding and protecting the DNA or RNA that's inside. So that's a generic viral picture. So here you see the contrast of a bacteria. So this theory, those evolutionists think that all these other structures were lost and what remained became viruses. Second group, smaller in number, think that viruses were the actual first thing beyond totally inorganic material, that they preceded bacteria. And so here you see the molecular structure of a very short segment of RNA, and that this is what led to the development of a more complex molecule of RNA that had the property of being able to act as an enzyme. So the R refers to ribose in RNA. So combining the two words, they came up with the word ribozyme, meaning an enzyme based on the ribonucleic acid. And this was able to catalyze other chemical reactions and start making more complex molecules. And so they imagine this collection of various whatever, whether it's a pre-ribosome, a pre-retrovirus, various protein molecules, which are, in this case, they're labeled as peptides, and that these things develop the ability to replicate themselves and eventually be transformed into DNA. But this is all wishful thinking. There is no evidence for this whatsoever. It's a great imagination is what it demonstrates. And we have to remember, it's very clear that even though we are told relentlessly by evolutionists that time plus chance equals information, that simply is not true, because matter cannot give rise to information. Matter is used to store information, but it cannot give rise to information. And so this is unfortunately something which is drummed into the heads of all of our school children, and many adults retain it as well. But this is simply not true. Time and chance does not create information. Well, I make that point because that's what they're saying when this RNA develops and then eventually leads to DNA and all the information that these molecules contain. Then the third idea that some evolutionists have is that viruses arose out of DNA segments that can move around within the total amount of DNA in a cell. And these are very specific types of segments that are called transposons, which means that they can move across, the trans meaning across, from one position to another position. And indeed, transposons do exist, and they're very important. Now, when they were first discovered, they were called jumping genes because it was assumed that these were random chance events that had no purpose or plan. But with further study, it has become crystal clear that these transposons indeed are extremely purposeful when they move from one place to another in response to a change in the cell's environment. And that these transposons, by moving to a new position, turn other genes on or off to alter what the cell is doing to be able to adapt to its new conditions, its new environment. Or another thought they have is that the viruses arose from these small circular segments of DNA called plasmids. They're not part of the chromosome, of the large circular chromosome you see on the left in the lighter blue color. But again, these are all just wishful thinking thoughts. There's no evidence for any of this. So they think that these things are the way that viruses came into being, evolving into a separate form of existence from bacteria. Well, let's take a look at the biblical model and how reality coincides with the history we have in Scripture. As I said at the beginning, I have to talk about bacteria because the interaction among bacteria and viruses is so basic and critical to the whole ecosystem. So I have to say a few things about bacteria here. It's estimated that there may be as few as 10 million or maybe as many as a billion different bacterial species. I don't think we'll ever know exactly how many. It would take forever to be able to examine all of them and sequence their DNA to figure out the different species. But the important point here is that bacteria are the base of life support systems. They're the base of ecology, whether it be in terrestrial environments, in land environments, soil. Bacteria there in the black square are the bottom, the foundation, so that larger one-celled organisms can eat them. And the bacteria thrive by decomposing organic material, either from dead plants or dead animals or just that's in the environment. And then larger organisms in turn eat, for example, here the protozoa or a fungus and on up the food chain. Well, it's the same thing in the aquatic environments, whether it be the ocean, lakes, ponds, streams, or rivers. But again, in the black square there, bacteria will decompose organic material, either from once living organisms or that's just dispersed in the environment, and in turn are eaten by larger organisms and larger organisms up to the food chain. Well, this is relating to viruses in that the viruses will use bacteria to reproduce themselves, to infect the bacteria and to be able to multiply their numbers tremendously. So the estimated number of viral species that affect organisms that are made of more than one cell, multicellular organisms, plants, animals, us, fungi, is about 100 million species of viruses that infect these types of organisms. And the estimated number of viral species that affect bacteria, we have no clue. It's probably much even greater than that. So there's this very close interaction among the viruses and the bacteria. Just for example, in the ocean, they found as of the writing of that particular article, approaching 200,000 species of virus in the oceans. So the next time you guys go to the coast, as you go swimming in either the Atlantic or Pacific, just think about all those viruses in that seawater that you're swimming in, and probably even getting some of it into your GI tract. But you don't have to worry about it because those viruses don't like our cells. They don't affect or infect us. And so that's not a problem. Well, so what is the biblical model here for the importance of viruses, knowing that they are so tightly interwoven with bacteria, both in land and sea? Well, the analogy here is think of the virus carrying out the job of taking DNA from one organism and transporting it to a different organism, much in the same way that a honeybee takes pollen from one flower to another flower to pollinate the second flower. So I think that's a beautiful and accurate analogy as to the role of viruses in the biblical model. And so the purpose of this would then be to be able to impart information into a new organism that didn't have that particular information to be able to deal with situations that it finds itself encountering. So it's now understood that this viral transfer of genes is critically important for organisms to be able to deal with the changing conditions. An example that's very common here in Arizona, and I suspect maybe even with the iron mines in Minnesota, since we have so many mines here of all the precious metals and many others as well, is that mercury used at the mine sites, or actually that's native to the ore that's there, is it toxic? But yet there are bacteria that have the ability to detoxify mercury. And so that's what you see here with this diagram, that mercury in form B, which they're showing as frowning and being toxic, it converted to A with this halo and smiling angel face. And so the bacteria that have the enzymes that are able to carry that out, those genes for those enzymes can be transferred by viruses from one species of bacteria to a different species of bacteria that lives in different conditions. And so in those other different conditions, and those bacteria acquire the ability to detoxify the mercury as well. So this is a very important role here for the viruses to be able to move that genetic information from one organism to another. Well, it's the same thing in aquatic environments. And you may remember that not too many years ago, there was a alarm put out that we should not be eating tuna that was caught in the vicinity of Japan, because the Japanese had been dumping tremendous amount of mercury from industrial sources into the ocean, and it got into the tuna and made them toxic to us. But yet there are particular species of bacteria in the ocean that are able to convert the form of the mercury so it's not toxic and is bound up and makes the ocean and the fish caught in that area of the ocean much safer for us to eat. So these viruses play a very important role in transferring that information of being able to deal with the mercury from one species of bacteria to another. Another very important example here, and there are many more, but this is one of my very favorite, is this particular bacterium, Alcanovorax borcumensis, is one of quite a few different species of bacteria that break down oil in the oceans. This from, let's say, from the oil spills that happen today, either from a tanker or from a oil rig that crashes and has a problem. But these bacteria have been doing this for quite some time, probably since the time of the flood, because natural sources of oil in the Earth's crust seep oil into the ocean, And then since oil is lighter than water, it's less dense, then it then rises and floats to the surface. And so since at least the time of the flood and probably all the way back to creation, there have been bacteria of which this is one species that are able to break down the oil molecules. And it's called alkanovorax, vorax meaning eating, like omnivore or herbivore, carnivore. And alkane refers to these oil molecules that are strictly made up of carbon and hydrogen atoms, nothing else other than carbon and hydrogen. So these bacteria are able to break them down. And so when that huge problem arose in the Gulf of Mexico some years ago, you know, the alarmists were saying, oh, this is the end of the world. And, you know, the Gulf of Mexico is never going to recover and it's going to be a total disaster. Well, those bacteria got busy chewing up on that oil and multiplying like mad. And then viruses were, you know, able to be used to transfer that information, that genetic information, to other species that also would be in maybe different parts at different depths or different temperatures of ocean, depending upon depth, and also be able to deal with the oil there. So those bacteria are very happily cleaning up these oil spills, thanks to the viruses being able to transfer that information from one species of bacteria to another. So here's a quote, and the caption at the top of the slide, the title of the slide says creation model viral viewpoint, but yet I need to emphasize that the source of this quote, University of California, Davis, is a totally secular source. So I don't mean to imply that Dr. Sivanen is a creationist. Most likely not, since he's on faculty at UC Davis. They probably wouldn't allow that. But the point being is that from a secular source here, we have this point of view, which is consistent with the biblical point of view. And the quote is, if viruses were nothing but bad news, you would expect cells to be taking great pains to evolve resistance to them. And they do express resistance in some way, which we're going to talk about. But they also seem awfully accommodating in others, and we're going to talk about that as well. So in other words, cells have specifically designed receptors to accept viruses. Now, I made the point before that chance events and time do not cause information to happen. So I just want to emphasize that mutations cause either mutations in DNA cause either no change or loss of information, never a gain of information. Now, it's possible there may be a gain of function due to loss of information, such as the brakes on a particular molecular pathway being lost, and therefore that pathway now runs at full speed and does things that it was inhibited from doing before. So there can be a gain of function, but never a gain of information. And evidence is indeed accumulating that most, if not all, harmful viruses and bacteria are mutated forms of non-pathogens. Pathogen is something that is disease-causing. And so this is consistent with everything being perfect at the time of creation until the fall. And then after the fall, with the curse on the whole universe due to the disobedience at the fall in chapter 3 of Genesis, that then mutations started developing, and things that were not harmful initially became so. So the idea here is very consistent with people who have experience with dealing with trying to program computers. If you put garbage in, you get garbage out. So when there are mutations in the DNA or even the RNA, then things do not work well. Same as if you have as we use the term, virus or a bug in a computer program, then it does not work so well either. So an example of this is cholera, disease cholera. Uh, it's been established that cholera, and that's the full scientific name for it, the genus and species, Vibrio cholerae, was once harmless, but became toxic after receiving the CTX gene, most likely by viral transfer. So cholera, its initial and still positive purpose is to break down the shells of crustaceans, of crabs and shrimp and other hard-shelled organisms. Because just like if trees accumulated on land when they died and weren't broken down by termites and bacteria and fungi, the land would be covered with dead trees. Well, same thing in the ocean. These shells need to be broken down and the materials in them recycled, and that's what the Vibrio organism does. So in its place, it does an excellent service and is beneficial. But if it's in the wrong place, such as contaminating food or drink for a person, then the disease is deadly. And what happens is this gene causes a change in the operation of the cells that line the GI tract, the gut, and these cells start pumping the chlorine and the sodium out of the cell into the gut itself, and the water follows that due to osmotic action, and so the person within hours can dehydrate and die unless very rapid IV therapy is initiated, and then bacterial therapy to kill the bacteria, and antibiotics to kill the bacteria so that the whole process can be brought to a halt. So this is an example of how viruses can transfer something and cause information to be taken from one organism to another. So all of this does fit the creation, corruption, meaning fall, catastrophe, the flood, and confusion, the judgment at Babel model based on biblical history. And then to finish that out, it would be then Christ, the cross, and then consummation when the current universe is burned up and the future perfect universe is then established. So I'd like to contrast here the two models. The evolutionary model says that starting from a very small gene pool, as in that first unexplained bacteria that came into existence, which they never will be able to explain, increasing due to mutations and selection. And again, they say that's by a gain of information and changing from one kind into another kind. But I've already explained how chance events in time never ever cause a gain of information. So this model is simply not following the laws of science, and it's actually anti-science. And it's built on a house of cards, which is really actually already fallen down. I consider evolution really to be a dead man walking. On the other hand, we have the creation model that in contrast starts from a very large initial gene pool created by God in creation week, a little over 6,000 years ago in normal 24-hour days, dividing by migration after the flood, after the organisms left the ark, and programmed filling. And that involves at least 5 different mechanisms, which we won't go into today. That's a whole different talk. But an ability to either have information transferred or to have shuffling of information within an organism. And we're going to talk about that shuffling in a bit here. So that you get changes within a kind, such as from the pair of dogs that came off of the ark, we now have 34 species, or from the pair of cats, we have 36 species, or the pair of the horse kind, 8 species, or the pair of the bear kind, 8 species after the flood. So that's that shuffling or loss of information. within a kind, not change from one kind to another kind, as the evolutionary model would like us to believe. And this has as its foundation the history given to us in Scripture, and we can rely on that. It never changes. Let's take a look a little more at viral anatomy. So I mentioned those 4 basic shapes of viruses: the cylinders, the 20-sided figures, the spheres, and the lunar landers, the bacteriophages. So the top row are the diagrams, the bottom row are actual electron micrograph or electron microscope photographs. And those are always black and white. So these are colorized just to make it easier to see the contrast of the various structures within the virus. So these are colorized transmission electron micrographs. So the basic viral structure is the RNA or DNA inside. The term that covers both of those together is nucleic acid. And then that protein coat, the capsid, the protein coat. And you see there it says naked nucleocapsid virus, meaning that it does not have an envelope. It's just simply the protein coat. And the protein coat is made up of individual protein molecules that form the structure of the capsid as they are combined with hundreds and thousands of individual molecules to make the coat. The syllable mer, M-E-R, means segment. So capsomeres means the segments of the capsid, each individual protein molecule. Now here it shows an enveloped virus, which virus, dotted virus that came out of a cell that was infected, also known as a virion, daughter virus virion. As it exited that host cell, it took with it some of the membrane from the host cell, and that became the envelope. So the origin of the envelope is from the host cell, not from the virus itself, but the virus acquires it from the host cell. So that envelope initially has protein from the host cell, but those become replaced by the viral proteins to a great extent, or even maybe totally. So here in this diagram, you see that inside the core is the RNA or DNA, in this case, RNA of the virus. And then there is that protein coat, and you see some spike proteins here. And then there is the envelope on the viruses that do have that. And that envelope comes from the cell membrane of the host cell. So that is a double layer of molecules that are lipids, are a type of fat molecule that in the double layer make up the envelope. So our cells have that lipid bilayer to define what is in from what is outside of the cell. And then here it's showing viral proteins that are in the form of spikes. Not all of them are spikes. Some of them are, and some of them are just the basic coat. So what are the functions of this capsid, this viral coat? Well, it protects those nucleic acids, either the RNA or the DNA, and it attaches a particular type of protein, attaches the viral DNA or RNA to the suitable host cell. And that category is called heme agglutinin. So agglutinin means like to stick to, and heme refers to blood. This is where it was first found. And so this is very necessary for the virus to be able to dock with the host cell. And this is very specific. So I'm using this illustration here of the International Space Station, and with a spaceship coming from the surface of the Earth to the International Space Station. And you see there that docking mechanism, it has to be exactly, precisely the same for the two parts to match and to be able to form a good union, and then for the people to transfer from one of those two objects to the other, whichever direction they happen to be going. So this is a great analogy, is that the virus has to dock with the host cell so that the DNA or RNA in the virus can then be injected into the host cell. So that's what that's about with the hemagglutinin for docking. And then that viral material, DNA and RNA, then is injected into the host cell. And then other proteins here in that viral coat, in that capsid, stimulate our immune system, or the immune system of a plant or animal, to produce antibodies to fight the infection, to help protect the host and do battle to be able to recover from the infection. And then there is a class of proteins called neuraminases that are responsible for detaching those daughter viruses, the virions, from the cell as they exit. So they have to be able to be clipped from the membrane as they exit. And that's what neuraminidase does. That's its role. So that they can become free to then to travel and infect other cells as well. Now, in that capsid, in that protein coat, are other enzymes as well. And in the RNA viruses, remember I said some viruses are DNA viruses, some are RNA viruses. In the RNA viruses, there is a particular critically important enzyme called reverse transcriptase. So let's break this down. Script refers to writing, trans meaning writing across, ase meaning the enzyme that writes across, and reverse meaning the enzyme that writes across in a backward direction. Meaning, instead of creating RNA from DNA, it creates DNA from RNA. And the reason for that is, in the host cell, the information from the virus has to be in the form of DNA to be incorporated into the host cell DNA. So that's why the reverse transcriptase exists in the RNA viruses. So what you see in that bottom left part of the slide there in the gray diagram is the cell has already been docked by the virus. The genetic material, the RNA, has been injected into the cell. So you see that first wiggly line is the RNA. Then the reverse transcriptase converts it into DNA so that DNA can be incorporated into the host cell machinery so that it can be copied and multiplied greatly. And also a class of enzymes called polymerases, which are the ones responsible for the copying of the DNA, making many, many copies from the original, in this case, RNA converted to DNA that's now in the host cell. So the viral polymerases literally commandeer the host cell machinery. They take it over and say, okay, your factories are not going to make what you normally make for yourself. They're now going to make what the virus needs, what the virus's RNA converted into DNA determines you will make. So there's been an appropriation of the cell machinery. So these viruses that contain this reverse transcriptase are called retroviruses. So here you see the picture of the virus, the bacteriophage, looks like the lunar lander, and here's the retrovirus. So here you see the release of the enveloped virus in diagrammatic formation. As it leaves, it takes part of the cell membrane from the host cell. The neuraminidase has been utilized to separate the daughter virus, the virion, from the host cell. And now there is an example of a virion, the daughter virus, that has exited from the cell. And here, again, neuraminidase is that enzyme that is necessary for that separation to occur. And this is a colorized electron micrograph showing an infected cell with multiple copies of the virions, the daughter viruses, either having already exited or in the act of exiting or getting ready to exit from that host cell. And when this occurs enough times, the combination of these virions leaving with membrane and with the cellular machinery not being able to make its own products, but rather just making the products for the virus, then the cell dies and ruptures. Well, let's talk a little more about these receptors. We have many, many different kinds of receptors on our cell membranes. And here you see 8 different examples of different types of molecules that have their own specific receptors, and they have particular actions occur as a result of those initiating molecules docking with their specific receptors. But there also are these receptors for viruses, and they are quite specific. So the virus encounters the susceptible host cell. It absorbs to that specific receptor site on the cell membrane. And because of the exact fit required, viruses have a limited host range. So here's an example of a coronavirus, and has its specific receptor there, as you see in the diagram, or an adenovirus, which is one of the causes of a common cold, with its specific receptor on its host cell. Well, here are examples. For example, the parvovirus affecting dogs, or an example for papaya virus affecting these leaves on the papaya plant down in Mexico, or the swine flu virus affecting pigs, the avian virus, bird flu virus, for example, affecting geese, tobacco mosaic virus, dealing with tobacco plants, or I have here the business of the bat and the coronaviruses that affect it. Or it can even be even more specific to a particular type of tissue only. So for example, in our upper respiratory tract, in the throat, or even the upper part of the airways and of the lungs, and in our nose, there are particular viruses that only have the docking mechanisms that match the receptors in those tissues. And so, for example, rhinovirus is another common cold cause. A rhino meaning nose virus for the common cold here. Or we have the example here, these are liver cells, healthy liver cells, and they have the receptors for the hepatitis C virus. So that's how we get that type of hepatitis. Another example would be here, the lining of the gut, and it has the receptors for, among others, the norovirus causing diarrhea and possibly some vomiting as well. And then this is brain tissue, this is central nervous system, brain tissue. And in those cells on those cells are the receptors, for example, with the Zika virus, which I'll show you at the end here. So there's quite specificity for these things. So here again is just the overall sequence of events. The virus arrives at the future host cell. It has the appropriate match of docking mechanism with receptor. it does dock, and then the DNA material is released into the inside of the cell, and those enzymes take over the cellular machinery of the host cell so that the viral DNA is reproduced, whether it be a DNA virus or an RNA virus that has the enzymes, the retro enzymes to convert the RNA to DNA. So the DNA is then incorporated into the host. Messenger RNA is made, which is then responsible for making the proteins that are needed for the protein part of the virus, and as well as the viral DNA or RNA. So then those parts self-assemble. It's amazing. They self-assemble into the properly organized daughter viruses, the virions, and then exit the cell, sometimes taking the membrane with it to have the envelope. So about 3 to 100,000 virions, daughter viruses, may be released from a cell. And this can happen as rapidly as 8 hours or maybe even 36 hours in length of time. So in the course of at most a day and a half, you can have 100,000 viruses from that one cell. It's astounding. Well, so how does the human immune system respond to this whole situation here? Well, these are the different types of cells in our immune system. I'm just going to focus on 4 of them here, because these are the ones that are most often the initial ones that encounter and deal with the invading element. And I'm going to focus on just one of them as an example here. I'll tell you that the dendritic cell is in your skin. And again, these are the guardians in the immune system. The microglial cell is in the brain, the central nervous system. Macrophages are out roaming through all the tissues everywhere looking for who doesn't belong there. And the B cells are in locations ready to respond to the triggers that occur when the invaders show up. And then I want to focus on just the example of the microglial cell here, showing how it responds to that thing that doesn't belong there. So it changes shape, it engulfs the invader, digests it, and then presents parts of the invader to the B cell so that the B cell can then process it and then convert into either a memory B cell, so that in the future, the body will already know the makeup of this invader and will have the factory ready to roll. So like the factory is going to be put into mothballs. So if that particular invader shows up again, that factory can be immediately put into gear and have a more rapid and a much stronger, higher response on the second exposure to that same invader. And then some of the B cells also then convert into plasma cells, and those are the actual factories that make antibodies. So this is the basic structure of an antibody. This is what does the work of binding to the invader so that it can be immobilized, taken out of action, and quit causing problems for us. And so the constant region is the same in any antibody, because its job is to be the connecting portion, either connecting to a cell or just connecting the 2 variable regions, which will then interact with the invading agent. And those variable regions are variable in that, meaning they're different depending upon which plasma cell makes them. One plasma cell will only make one kind of antibody, and a different plasma cell will make a different kind of antibody. So that's what the variable means. It doesn't mean that one cell will make different kinds of antibodies. So again, the specificity is critical, and it is maintained for a particular plasma cell, a particular factory. That factory will only make that one kind of antibody. So you see here the label DNA. So this is a segment of DNA, and this is very simplified, but just to get the concept across. So you see there the plasma cell is the factory. And there are sections of the DNA that are spliced together, and there are sections of the DNA that are spliced out. The exons are the ones that are spliced together. So you see they are numbered 1 through 5. The introns are the intervening portions of DNA. So they are not used to make the protein, but they are used to do many other very critically important things. So you see here, this is the resultant RNA made from the DNA. And you see the 1, 2, 3, 4, 5 with the introns in between in green. And then you see how there is a different way of splicing them together. So for example, in the left lower segment, it's 1, 2, 3, 5. And the right lower segment, it's 1, 2, 4, 5. So these can make different types of antibodies. So this is how we can have a small amount of genetic information make a huge number of different kinds of antibodies. So you see these are different from each other, with 3 different than 4. And again, this is very oversimplified, but it gets the concept across. And all these other intervening segments, the introns, make other kinds of RNA, about 30 different kinds of RNA, which are critically important for regulation of what's going on, of how much is made in what time, what circumstances. For example, change of temperature, that they can alter what's going on here. So those other things are not junk, they're not useless, they're not leftovers of evolution. They have very important programmed uses. So how many different kinds of antibodies can we make? Well, take this concept of the introns and the exons, the parts that are spliced together and the parts that are spliced out, and then the different combinations, how they can be spliced together. And imagine a slot machine with many more segments to it than this. And it's estimated that we make about 10 billion different kinds of antibodies. Now, I wish I were with you in person so I could see your reaction to this number, because this is absolutely astounding. But this is critically important because we have to have these antibodies that match everything out there, every kind of bacteria, every kind of virus, every kind of fungus, so that we have those antibodies, the capability to make those antibodies for anything that's out there. So this is an absolute marvel of design, of engineering, so that a relatively small amount of DNA can make such a huge number of different kinds of antibodies. This is something that had to have been designed by our creator savior. Nothing like this could ever evolve. So here's that shape of the antibodies with that constant region and then the variable region out at the ends of the forks of the Y. And these 3 types are free floating in the bloodstream: IgG, IgD, and IgM. IgM has 5 of the units combined together. And then in the IgA form, 2 of these units are together, and the ones that are in the IgA classification are in secretions, such as either in mucus to help protect against infection in the airway, or in the gut, mucus in the gut, or in breast milk, for example, that type of secretion. So IgA antibodies are in breast milk, so that that newborn babe has passive protection supplied by mom until the baby starts developing its own antibodies in response to being exposed to the outside world. And then IgE are attached to cells And unfortunately, these are the ones that are responsible for allergies and, having eczema, asthma, this kind of stuff. Those are responsible for those overreaction situations. Again, everything is very specific. A specific antibody in blue responds only to a specific part of a virus or bacteria or fungus or peanut or cantaloupe or whatever the person may be allergic to. And so you see their specificity of shape is the interaction there, just as it is with the docking mechanisms for a virus to enter a cell. So we have this standing army ready to be mobilized when faced with another exposure to that same organism we were exposed to earlier, because of those plasma cells and memory cells ready to roll. So that's how the body responds to an infectious agent. Well, let's take a look at how viruses can be very, very valuable to us. So I mentioned bacteriophages are the ones that look like the lunar landers, and that means bacteria eater. So here you see this bacteriophage riding herd on the bacterium. And this is electron photograph, micrograph, showing several of these bacteriophages attacking this bacterium. So after docking, the DNA or RNA of the virus is injected into the cell, and then it makes its way into the nucleus so it can take over the machinery and have its copies made. So here is attachment in the way of review, then the injection of the DNA or RNA, okay, then the takeover of the cell's machinery so that the virus's proteins and RNA or DNA are copied, and then they self-assembly, and then they exit. And after enough of this has happened, then the cell lyses, meaning it breaks open, it dies. And then the process repeats itself as new cells are infected. Well, here's a very interesting way in which work is being done to see how we can use viruses to our benefit. So these bacteriophages, the viruses that attack the bacteria, it's being looked into to see how we can use them to treat bacterial infections in our bodies. And because of the specificity, those particular viruses are not a threat to us. So they are specific for that particular bacterium. So they're non-toxic to our cells, whether it be in skin, in the GI tract, or the respiratory tract. Non-toxic for the bacteria that we have in those locations, non-toxic for our cells, all of our cells. And there's no antibiotic involved, so there's no risk of allergic reaction to antibiotic. So this is what's being looked into now, as to how we can employ bacteriophages to treat bacterial infections in us. One study going on in Mayo Clinic is in a surgical situation where somebody has had an artificial joint put in, say a hip or a knee or any other joint. If currently or historically, if a bacterial infection has occurred after the surgery, then the surgeons had to go back and reopen the wound, open up things back down to where the joint was put in, wash it out thoroughly, close it back up, and put the person on antibiotics. Well, it's a shame to have to undo and redo the surgery that was performed initially. So if there is going to be success with these bacteriophages to kill those bacteria, then the surgery can be left alone. The joint does not have to be reopened. And that would be marvelous, especially in the situations where antibiotics do not kill those bacteria, that we can use viruses to do so. So that's work that's going on right now. So this has great potential. It's an amazing way to think about how to do things. Another way in which viruses are very valuable for us is in curing genetic diseases. So in downtown Phoenix is a world-class research facility called the Translational Genomic Institute, nicknamed TGen. And this is on the biomedical campus in downtown Phoenix, where there's a medical school with various universities working together with colleges of medicine, nursing, physical therapy. There's a cancer research facility. There's all sorts of stuff going on there. Well, the task of TGen is to take the information we have in knowing the sequence of the genome, and then using that to see if normal DNA can be given to people who have mutations in their DNA, inherited diseases, genetic diseases, so that instead of just treating the symptoms of the disease can actually be cured by inserting the normal DNA into their cells. Well, part of this thought process is relating to the question, why aren't bacteria already wiped out by viruses? As I mentioned much earlier, they do have means of protecting themselves to a degree from viruses, and that means is called restriction enzymes. So what are restriction enzymes? Well, first of all, they are a gift from God, and used restriction enzymes to put human DNA into bacteria to make products that are human in result. And so the term recombinant means combining DNA from two different sources, different species. And so these enzymes, these restriction enzymes, protect bacteria by slicing and dicing the viral RNA or DNA. That's the role they serve in the bacteria. But we use these things so that we can use these restriction enzymes to cut out a piece of our DNA and insert it into the bacteria and use the bacterial machinery to make the human gene product. So you see here that it says that these enzymes cut DNA at places that are palindromic. Well, what is a palindrome? It is for example, in an English sentence, the same read left to right or right to left. So whichever direction you read this, it still says, do geese see God? Well, that's an analogy for stretches of DNA that are palindromic. You read it in either direction and it reads the same thing. So here you see, as you look at this, that it's the AATT at the top row going from left to right, and in the bottom row going from right to left, AATT, and then so on. So this is a palindromic section of DNA, and this is the type of site at which a restriction enzyme will slice open the DNA. And different restriction enzymes will slice open different palindromes, different sections of DNA that read the same in both directions. So we use these so that we can cut out a human gene and put it into a bacteria. Now you see here where it's AATT in either direction, that is a single-stranded extension, and that's called a sticky end, and that's how the human DNA is inserted into the bacterial DNA so that everything can be perfectly matched up and then sealed and not fall apart. So here is in diagram form what I've been mentioning here. So this is a section where there's a palindrome, and this particular restriction enzyme will recognize this particular palindrome. So there you see where there is slicing out from the one source. That's going to be the recipient. Then you see here in the donor source, that same enzyme is used, and so it slices open at the same sequence, and then the two are combined, and then an enzyme is used to seal the seams, so to speak, in the analogy. And so now that human gene is in the bacterial DNA, the host DNA, in this case, the bacterial DNA. And so here is an example of how we use this. When I was first in my early years of practice of medicine, diabetics had to receive insulin from pigs and cows harvested from the pancreas of slaughtered swine and beef because we had no way of obtaining human insulin. Well, it did the job, but unfortunately, the problem with the porcine insulin or the bovine insulin from cows is that those were foreign proteins, and the immune system of the person recognized those as foreign proteins and would develop antibodies. And so with time, that diabetic's dose of the bovine or porcine insulin had to be increased to higher and higher and higher and higher doses. Well, that was not optimal. But then with the advent of all this technology, that problem was solved. So here you see the insulin molecule is made up of two short, very short protein segments. And so there's the DNA for each of those two segments. Using the restriction enzymes, those genes were sliced out of a human cell, pancreatic cell, and then inserted into the circular DNA inside the bacteria. The bacteria multiply like mad, producing zillions of molecules of the two chains, A and B chains of insulin. So those were then harvested and purified, and then the A and B chains were put together, and they would naturally combine to form the full insulin molecule. And so that's how we ended up with the ability to prescribe and administer human insulin to diabetics with zero problems, no foreign proteins, no reactions, a good stable dose, and using with E. coli, these bacteria, is a pretty inexpensive way of doing it. And certainly a lot easier than harvesting insulin from the pancreas of cows and pigs. Marvelous gift to be able to do this. And that's because of these restriction enzymes that the bacteria have to protect themselves from viruses. Well, we can do this not only to have bacteria make different things for us, like human growth hormone or insulin or other items. We can also use viruses as a vector, as a means of putting the normal DNA into a person who has a mutation in their DNA causing a disease. And so you see here that a gene by a harmless adenovirus, the one that's would cause the common cold if it were able to, but this is a harmless version of it. So that the gene is put into that virus for whatever the particular disease is. And then the virus delivers the gene into the cell. Then that cell that's been targeted will now make the proper protein so that the cell will make the protein that it wasn't making before, or it will now have a receptor that it didn't have functioning before, so that whatever needs to enter that cell and was unable to before, now is able to, because that protein receptor now is normal and can function. So this is amazing stuff, and this is real life, real time stuff. This is what TGen is doing right now, and other institutes like it around the world. This is not pie in the sky. This is happening now. So we use these viruses, these bacteriophages, we say phage for short, to deliver drugs and to deliver genes, so that there is chemical modification, genetic engineering, attacking cancer cells. And so disease can actually be cured at the molecular level rather than simply treating symptoms. And so it's these viruses that make this possible. Well, let's talk about just 6 vicious viruses as we near the end here. So the doc is saying to the patient who has that giant bacteriophage on his back, look, if you had a bacterial infection, I could prescribe what you're asking for, an antibiotic, but it's clearly a very painful virus. Well, if I were with you in person, I would ask you if you could think of what a very painful viral disease is. But since that's not really easily done, I'll just go ahead and show you that these zoster virus causes shingles. And that is exceedingly painful. That pain is horrendous. And the problem in diagnosing early on is that the pain is present before the rash becomes evident. And this works by, say in childhood, someone got chickenpox, that virus becomes dormant and resides in the dorsal horn cells of the spinal column, spinal cord. And usually in older years, although it can happen even in the 30s or 40s, but usually it's in older folks, some form of stress, and it can be any form of stress that's significant, causes the virus to reactivate. And so then it multiplies and travels along those nerve cells, causing tremendous pain and then a subsequent characteristic rash. And so there you see that geographic distribution of the rash following the distribution of that nerve cell from the midline, from the spinal cord, along as it curves down and around towards the midline in the front. And the rash can be at multiple levels, multiple nerves involved, and it could even be bilateral if nerves on both sides are involved. It can be a very debilitating situation and extremely painful. Well, another very problematic virus, very vicious, is the human immunodeficiency virus. And the reasons it's so terrible is because it attacks the T cells themselves in the immune system and wipes out the immune system. So here you see electron micrographs, colorized, of the virus entering, being in the cell and then exiting the cell. And then, of course, there are multiple copies, thousands of copies in each cell, and then it goes on to other cells, so that with enough time, the immune system itself is wiped out, and then that person is open to infection by anything, including other organisms, viruses or bacteria or fungi that normally do not cause infections in people. And that's why this is so devastating. So it wipes out these T cells. It also can cause certain types of cancer, in particular one called Kaposi's sarcoma. So it's a terrible thing. Zika virus is another terrible one that affects the central nervous system, the brain, has specific receptors there, and it affects babies as they're developing inside the womb, inside the uterus. And so you see there with the far left, you see the baby that has a normal head size. And then depending upon the degree of the effect here, the head is smaller and then really super smaller. And microcephaly means small head. And so these babies are born with a small head. And so you can see here that the proportion of the size of the head to the trunk is tremendously out of balance. And of course, this also affects mental ability as well. And it's irreversible. It's really a tragedy. This has been known for quite a few decades. It came out of Africa, but it hadn't been rampant until just a few years ago. It managed to be spread to Brazil, and there was a great outbreak there. And now it's even present here in North America. But it's important to keep these things under control. Ebola, many of you may remember the tremendous problem in 2014. There was a horrendous pandemic of Ebola in Western Africa. And this virus has an extremely characteristic shape to it. No other virus has this shape. It's called a filovirus, philo meaning long and skinny like a file. But what makes it unique is the way it folds up like this, giving this characteristic shepherd's crook picture. And it originated in bats in Africa as well. And again, it's been known for some time, but earlier on, an outbreak would occur and it would be self-limited and then disappear for a few decades and then reappear and then disappear. Not such a huge epidemic until this one in 2014. And it affects several different types of cells, including those in the immune system, the kidney, and the liver. And it just turns these tissues literally to mush, and people just bleed out from anywhere and everywhere, every body opening, every orifice in the body, and anywhere there's a needle stick or any kind of a wound. And these people just bleed out, and it's a terrible, terrible, terrible way to die. And that's just an example of bleeding in the skin, under the skin. There are many far worse, horrendous pictures, but I chose not to use them in this presentation. And here are workers in their hazardous material suits, their spacesuits, they nickname them, to protect themselves as they take out a person in a body bag so it can be buried and or burned. West Nile virus, you see here these spheres, transmitted by mosquito. And in the darker green areas, it's more prevalent. The darkest green is affecting people, the lighter green, mainly birds. And many millions of birds have been affected by it as well. And so here you see the distribution, and you can see that, well, they don't show any green splotches in Minnesota, but in your neighboring states, you can see even occurrence in those locations as well. But the prime region is down here in the Southwest, especially southern Arizona and southern California is the primary region for it. And so we're a little more skittish about it here. And it affects the central nervous system. Fortunately, very few people have died from it, but it can leave lasting deficits in the central nervous system if a person recovers from it. So we're pretty sure it's the West Nile virus, the doc says. And then the Epstein-Barr virus, many of you probably have dealt with it in the form of, or know of someone who has, in the form of mononucleosis. Typically happens in teenagers, nicknamed the kissing disease. So this virus causes a flu-like picture often, but it also, not always, but particularly more in blacks, will cause what's called Burkitt's lymphoma, a solid tumor. And the mechanism here is that the virus causes two chromosomes, number 8 and number 14, to break in a very characteristic place. And those two portions of those chromosomes translocate. They move to the position of the other broken part and then recombine with the other. So you can see here a part of chromosome 8 is on 14 and a part of 14 is on 8. The Q refers to the long arm of the chromosome. P refers to the short arm of the chromosome. And so with this break and transfer of the two chromosomes, there is loss of control of the formation of these lymphocytes, part of the immune system, and they multiply like mad and form these solid tumors. Well, there are lots of terrible things that happen with viruses. There are great things that can happen with viruses, such as using the restriction enzymes and using them as means to put curative normal DNA into patients who have genetic diseases. But most commonly, they're known for causing great sorrow, great tragedy. And so that's why I brought up Revelation 21:4. And God will wipe away every tear from their eyes. There shall be no more death, nor sorrow, nor crying. There shall be no more pain, for the former things have passed away. So even though there has been tremendous loss of life by viruses throughout the millennia, and even today with the people who have died because of COVID, we're to remember that those of us who know the Lord have the certainty that in the future we will be with him in eternity, where there will be no more death or sorrow or crying, and that things will be marvelously perfect. So we have that certainty to look forward to. It's not just a hope, it's an assurance that is for real, that we can count on. So that is the basics, I think, here with valuable and vicious viruses. So I don't know, I hope we'll have the means to take questions. We'll see if this works with the technology.
Could you speak to the COVID virus?
Yeah, what do you have to say about COVID?
Oh, I have lots of things to say about COVID. I'll just make some general statements. Unfortunately, COVID has been severely politicized. And what you hear in the press and the major media is not really an accurate reflection of what's really been going on. One of the important things to understand is, I looked at the CDC, the Center for Disease Control Statistics, myself. There is not this huge amount of excess deaths that we were told occurred, according to their numbers. Another thing that comes out of looking at their numbers is that what happened was that anybody who tested positive for COVID was coded as a COVID death, even though they didn't die from the virus. So for example, this is a real example, but it's a more extreme example, is a 25-year-old fellow who tests positive for it, but who dies from his motorcycle accident, was still coded as a COVID death. But this also happened for people who died from other disease processes, not trauma. And so when you look at the numbers, comparing the numbers for 2020 compared to the numbers going back to 2014 on every year through up to including 2020, all of a sudden people are not dying from chronic lung disease, from pneumonia, from coronary artery disease, instead, because they're being coded as dying from COVID instead of what actually killed them. And so it's very unfortunate that the politicization of this virus has caused such misrepresentation for political purposes. Now, I don't say that to minimize those who did die from the virus, but I do have to say that I think many of those deaths were not necessary, because the things that were very inexpensive and very readily available to be able to treat people were not employed. And so I think it's very important for you all to know that you should make sure that you have adequate vitamin D in your system, for example. Now, we manufacture our own vitamin D by being exposing our our skin to sunshine. And that we manufacture our own vitamin D, but also a faster way to get those vitamin D levels up. And it's especially more important for people in the more northern latitudes, such as you folks in Minnesota, is to take initially 10,000 international units of vitamin D daily for about 3 weeks or so, and then back off to 5,000 units in the international units per day. Now that's a lot of vitamin D, but it really dramatically decreases the incidence of people being able to get sick from the disease. In other words, it helps them resist the virus. And if they get sick, it makes them much less ill, decreases the severity of illness, and it dramatically decreases the number of people who need to be admitted into the hospital or put onto ventilatory support or die. It's amazing. And there have been nearly 2,000 studies around the world, and the results are uniform, and that there is a tremendous, tremendous benefit from vitamin D. And that's not the only thing that can be done. See, when you think back to all these people who were in the nursing homes who were dying left and right, well, and when you're in a nursing home, do you ever get exposed to sunshine? And the answer is no. You're stuck in a room and your vitamin D levels are low. So in addition to vitamin D, it's very important to have good levels of vitamin C, which you can get from eating citrus, drinking orange juice, or other forms of citrus. Strawberries and kiwi are also high in vitamin C. Or you can take pills for vitamin C if you want to go that route. So these things are very helpful. In terms of antibiotics, you remember that President Trump had mentioned hydroxychloroquine and Zithromax, Z-Pak, and the press just crucified him for saying that. Well, it turns out he was correct. He had good sources telling him that those indeed are effective. Another one that's very effective is ivermectin. And so if these very inexpensive, very commonly used and used for many decades antibiotics, now they are, primarily for treating bacterial infections, but they do have a certain degree of antiviral effect. And they do greatly improve people's ability to deal with the virus, especially if the ivermectin is started when they first start developing symptoms. So these types of things are the type of information that our health bureaucracies should be alerting everybody to. But they are very slow to do so, if at all. So it really pays to look at what's going on in some other countries around the world where they are doing these things and seeing the benefit that they're getting. Another thing is, I would hesitate to tell people under the age of 30 to take the vaccine. There are much higher incidences of problems, especially inflammation of the heart muscle, especially in males under 30. But if somebody is more elderly, especially in the 70s and 80s, and they have other diseases, a term is comorbid disease, then I would say, yes, the vaccine is a good idea. If someone's pregnant, I would say, no, probably should hold off because there seems to be an increased incidence of spontaneous abortions, miscarriages that occur with the vaccine. So, as with every other vaccine in the past, well, I should say in contrast to every other vaccine in the past, there's always been a multi-year observational period and trial situations to see what really happens before they're finally unleashed on the general public. That's not occurred this time. And so as time marches on, we're finding out more about the side effects with the vaccine. And so for a disease that is 99.9% non-lethal, or maybe 99.5% non-lethal, I think it is a crime to force people to take the vaccine. I like the idea of, the people who favor abortion, they always say what? Well, it's my body, so it's my choice. Well, I think we can use that same phrase ourselves. There's lots more I could say regarding the political end of it, but I'll hold off. If you have specific medical questions, I'll try to answer them for you.
Ross?
Yes,
I was talking to a biology professor, and I had read that they cut apart a dead person who had died, and they found that, I can't remember what it was it the virus or the antibody or whatever, was in every part of his body, and the biology professor said that was a good thing. I'm sorry, I can't remember what the word was that the article was, but it was apparent after they dissected the person after death, and it was all over, intricate in his body.
so the virus is all over, or the antibody?
That's what I can't remember. I'm so sorry.
Well, that would make a difference. Did you hear that?
Yeah, repeat the question.
Yeah, I'm not sure I understand it. An autopsy showed it's either the virus or the antibody all over the body.
Well, having your antibodies everywhere is good.
Yeah,
That's why we make them, is to fight infections. Having the virus everywhere is bad.
yeah. Okay, another question.
I've heard that the vaccine is not technically a vaccine. Can you explain that if you found it, or?
He has heard it. The vaccine is not technically a vaccine.
Well, I think we get into word slicing and dicing, and I don't care to say anything about that.
Yeah, it's a different mechanism, certainly. The other burning question, yes.
You agree that Rand Paul and Fauci is a liar and the virus came out of the lab?
He's wondering if Fauci is a liar and the virus came out of a lab.
I think Fauci is, let's just say, self-contradictory. I believe he has a political agenda. I believe the virus is definitely came out of the lab, and that it has been genetically engineered to infect people.
As a follow-up to that question, oftentimes we're hearing now that we know that the narrative of it coming from bats is not true, that there's this gain of function study that they were doing. I wonder if that ties together with this transfer of the genes, and if Dr. Kezele could comment on how that might work out.
Gain of function.
Okay, is there a question about gain of function?
Yes.
Okay, well, I think that's kind of like what I was referring to before, where the information, the genetic information can be altered. So there is no new information, but a gain of function, such as for it to be able to infect people when it couldn't before. I don't think this virus just jumped all by itself from bats to people. I think it was engineered to do so at the Wuhan lab. There are quite a few virologists around the world who have looked at the data, and they've come to that conclusion.
On that cheery note, well, I think it emphasizes the importance of turning to God. And I think we just know in general, God allows disasters because he wants us to realize we cannot solve things on our own. And yet people can continue to believe that they can solve things, they don't need God, or that God isn't there. And we certainly need to pray that people will turn to the Lord while there's a chance. Well, we are past 9 o'clock, and I think that we'll probably call this meeting to a close, and thank you all for coming.
Thank you.
All right. Thank you. You're most welcome.